Morning to all. Topic for today's discussion is on dental plaque. So, what is dental plaque? So, before going into dental plaque, we'll know about the colonization of the oral cavity. We all know our oral cavity is colonized by more than 500 different species of microorganisms. Approximately more than 150 different species of organisms present at the same time in the adult oral cavity. So, what happens to the oral cavity? Because there is a harmonious relationship between these microorganisms and the beneficial organisms, disease process present there. But when there is any immune suppression or an immunocompromised patient, there is an attack or a battle between these pathogenic organisms and the beneficial species. That is how a disease process develops. So, going into dental plaque. So, what is dental plaque? Dental plaque, to define, it is a specific but a highly variable structural entity resulting from the colonization of microorganisms on the tooth, on the restorations, and other areas of oral cavity, composed of mucin, microorganisms, debris, desquamated epithelial cells, all embedded in a gelatinous extracellular matrix. So, how do you differentiate dental plaque from Materia alba and calculus? Materia alba is very similar to plaque, but Materia alba doesn't have an organized structure like plaque and can be easily detached from the oral surfaces by a water spray. So, what is calculus? Calculus is nothing but it is a mineralized form of dental plaque. What is the classification, or how do you classify dental plaque? Dental plaque can be classified based on its location, that is above the gingival margin, it is called as supragingival. Below the gingival margin, it is called as subgingival plaque. Supragingival plaque is also called as marginal plaque. Subgingival plaque we can further subdivide based on its location. If it is associated with the tooth, tooth associated. If it is associated with soft tissue, epithelium or tissue associated. Now, this epithelium associated or the tissue associated plaque have one more category called as unattached plaque, where the plaque surface or the plaque will not be attached to any of the surfaces. Instead, it will be seen as an unattached loose layer within the sulcus or within the pocket human. In this picture, you can clearly see the unattached plaque here. Okay, this is the tooth attached plaque, and this is the epithelial surface. So, what is seen on the surface is called as tissue associated or epithelium associated, and this in between this layer, it is called as unattached plaque. Now, site specificity of the plaque. So, based on the location, this plaque can cause specific disease. As we have said, the supragingival or marginal plaque can result in marginal gingivitis. Now, the subgingival plaque. Subgingival, that is tooth associated subgingival plaque, and some amount of supragingival plaque. They can cause calculus and root caries. Whereas epithelium or tissue associated subgingival plaque can cause tissue destruction, that is periodontitis. We can compare the dental plaque to that of a biofilm. What is biofilm? Biofilm is nothing but what you can see on the stagnant water bodies. For example, water in the pond. You have a slimy, greenish, yellowish layer on the surface. That is called as a biofilm. So it is actually a matrix enclosed bacterial population adherent to each other and or to the surfaces or interfaces, the definition given by Chandran. So this picture, you can see this plaque or the biofilm onto a deciduous tooth surface. Similarly, a plaque or a biofilm, basically a biofilm on a stagnant water body. So what is the structure of dental plaque? Plaque structure is very similar to that of a primitive circulatory system. That is, it has got numerous open fluid-filled channels running through it, and the bacteria will be present within this matrix. As we have said, the matrix is acting as a barrier in which the bacterial substances are concentrated and retained, and metabolic interaction takes place between different bacteria. So I don't know how clear this picture is. This is a picture of showing the structure of dental plaque with bacterial colonization. Now, composition. What is the composition of dental plaque? Dental plaque has got organic and inorganic matter. The organic matter is nothing but it is polysaccharides, proteins, glycoproteins, and lipid material. Lipid material is actually the membrane of disrupted bacteria or cells. Inorganic is basically minerals like calcium, phosphorus, with sodium, potassium, and fluoride. Now, the important aspect of this particular topic is steps in plaque formation. What are different steps? Initially, we have a first phase, that is formation of an organic or acquired pellicle and then transport of bacteria to the surface. Second phase, we have initial adhesion. Next, we have attachment. And finally, we have colonization and plaque maturation. So what is a pellicle? Pellicle is nothing but just derived from adsorption of macromolecules. What is the composition of pellicle? It has got lipoproteins, it has got proline-rich proteins, phosphoproteins, protein, enzymes like sialic acid, and these act as receptors. That is, pellicle which is formed on the tooth surface act as a receptor for bacteria to attach to it. Attachment or formation of pellicle. Next is bacterial transport to the surface. How is bacteria transported to the surface? It is by means of three different ways. That is random contact. That is also called as Brownian movement. Next we have sedimentation. And next we have active bacterial movement. So random contact is the random attachment of bacteria after transport to the surface, sedimentation, and active interaction of bacterial movement. Now, next important step is initial adhesion and attachment. Adhesion is by means of two types of forces. We have short range and long range forces. Long range forces are Van der Waals attractive force and electrostatic repulsive forces. Short range forces are hydrogen bonding, ion pairing, and steric interaction. So after the adhesion, now attachment. Attachment, as we've already said, we have pellicle of the tooth surface. This pellicle has receptors which are called as cryptotopes. Similarly, a bacterial surface also has particular proteins which act as receptors, and they are called as adhesins. So there is an attachment between adhesins on the bacteria and cryptotopes onto the pellicle surface. Finally, we have something called as colonization and maturation. This is by means of a phenomenon called as co-aggregation. What is co-aggregation? It is intrabacterial connection, or it is a cell-to-cell recognition of genetically distinct partner cell types. It was introduced by Gibson and Nygaard in 1970. So, I'll give you examples for primary and secondary colonizers, which has got this intrabacterial aggregation or co-aggregation. Primary are the first colonizing organism, basically Actinomyces species and Streptococcus species. Secondary, we have Prevotella intermedia, Prevotella loeschei, Capnocytotega, F nucleatum, and Porphyromonas syngivalis. And these are the examples, that is F nucleatum co-aggregating with Streptococcus, E. loeschei with A. viscus, and Capnocytotega with A. viscus. Examples for co-aggregation. So this picture clearly explains the entire steps or sequence of events in plaque formation. See the tooth surface, in which we have an initial receptor or the pellicle which is formed or attached. This pellicle has a receptor called as cryptotopes. Next is the primary colonizers of organisms. These have specific adhesins with which it attaches to the pellicle onto the tooth surface. After which we have co-aggregation, that is intrabacterial interaction. That is, primary colonizers will co-aggregate with the second group of secondary colonizers, and thus the microorganisms or bacteria keep on attaching with each other. Finally, the entire colonization and maturation takes place. From this picture, we can clearly make out this group of organisms, that is Fusobacterium nucleatum. It is a group of microorganisms which can co-aggregate or adhere with a number of other different species. So this is the entire event happening during plaque formation. We have two specific examples or types of co-aggregation, that is called as concave formation and test tube formation. Example for concave is co-aggregation between Streptococcus and filaments of Bacteriopneumonia macrotubular Actinomyces. Example for test tube brush or bottle brush formation is filamentous bacteria and gram-negative bugs. The last portion of this topic is on plaque hypothesis, or the concepts behind the formation of dental plaque. Initially, the first thought was on a non-specific plaque hypothesis, which stated that all bacteria are equally pathogenic. Okay, so the disease process develops and the immunity of the host decreases, and more plaque accumulation happens. There is no specific bacteria responsible for formation of disease. All are equally pathogenic. So once the immunity weakens, it can cause disease. Later, specific plaque hypothesis came into existence, stating that it is not about it's not that all bacteria are equally pathogenic, but there is a specific or pathogenicity in a particular specific bacteria which is responsible for causing disease. All plaque are not equally pathogenic. So, according to the specific plaque hypothesis, it was stated that AA, that is Actinobacillus actinomycetin components, is responsible for causing aggregation, that is responsible for causing aggressive periodontitis. So that is an example for specific plaque hypothesis, that specific or virulent bacteria causing specific disease. Then we had unified, then we had ecological. Ecological plaque hypothesis states that any shift in the ecological conditions like pH or humidity, all those things can interfere or can influence the formation of plaque. Then we had updated non-specific, and finally the keystone pathogen hypothesis in 2025, Haji Shengalis. This hypothesis states that keystone pathogen or P. gingivalis is the predominant microorganism responsible for causing disease process, and various mechanisms also has been put forward based on this keystone pathogen hypothesis concept. The last is about the bacterial complexes given by Sopransky. It is called as Sopransky's complexes. We have red, orange, purple, green, yellow complex organisms. In this, the red complex organisms are P. gingivalis, Tannerella, Forsythia, B. Forsythia, T. dendricola is responsible for bleeding on protein. Thank you.